Since time began, man has lived with risk, surrounded by the forces of nature. It would have been possible for man to avoid locating his plantations and settlements in the neighborhood of volcanoes, but he did not choose to do so. Anything buried by lava will be rebuilt again on the same spot. Floods. Nowhere on Earth does man avoid the banks of a major river simply because it might flood, and people who survive a flood stay on. Earthquakes. You could even be forgiven for thinking that regions which are repeatedly shaken are particularly desirable places to settle. And if a tornado with its trail of destruction narrowly misses you and sweeps away your neighbor's home, your fear will turn to thrill. But no one would think of moving just because of it. What is it that makes people expose themselves to danger and put their own safety at risk? They know the odds. They want to push themselves to the limit, to experience the ultimate kick, whatever that is. Other people endanger themselves simply by suppressing all thoughts of potential danger in their everyday lives. For example, there are a lot of people who do not know that they are diabetic. One day, at home or at work, they lose consciousness and fall into a coma. The cause, which may have existed for years, is diagnosed for the first time in the emergency room. And yet today, it would be a simple matter to diagnose diabetes in its early stages. Until the 1920s, the diagnosis diabetes was tantamount to a death sentence. In those days, bacterial infections were the most frequent cause of death. For centuries, the biotechnology required to brew beer or to produce bread, cheese, or wine had been common knowledge, but no one dreamt that within a few decades, this same biotechnology would succeed in conquering bacterial infections by means of antibiotics. Challenging the limits. Today, many of the bacteria which have become resistant to antibiotics pose a threat once again. The same with diabetes. When the pancreas of pigs and cattle proved to be a source of the hormone insulin, which diabetics lack, it looked as if the illness had been conquered. With the help of that insulin, diabetic patients were able to enjoy an almost normal life. But no one had considered that from now on they would pass their illness on to the next generation. Challenging the limits. Today, there are some 120 million diabetics. By the 21st century, basic substances produced from animals would no longer be adequate to meet the growing need for insulin. Challenging the limits may sometimes even extend the options beyond the borderline. The biotechnical production of insulin is regarded throughout the world as the second great milestone of biopharmaceutical research and production after antibiotics. This was achieved for the first time in 1982. Since then, the life-saving hormone has been available in sufficient quantities to meet the requirements of all diabetic patients. And because it is a human insulin, foreign protein allergies have become a thing of the past. Discovering ways of reproducing the body's own proteins which the patient lacks. This was the goal which research teams of Boehringer Ingelheim across the world set themselves. Molecular biology became one of the main areas of basic research. Once its nucleotide sequence is known, the molecular instructions for synthesis of a human protein can be isolated from human cell plasma using an ultracentrifuge. The fraction of the genes in question lights up in ultraviolet light and can be extracted. These human genes now have to be inserted into the genome of those cells which will be responsible for producing the protein. For almost 2 decades, scientists have known that single cells occasionally exchange so-called plasmids by means of a plasma bridge. These are circular mini-chromosomes, double-stranded rings of DNA in the form of a spiral. By means of a restriction enzyme, which functions like a pair of chemical scissors, the plasmid DNA can be accurately cut between 2 base pairs. The human DNA, which has been treated with the same restriction enzyme, can thus be fitted into the plasmid ring. The adhesive of the genetic engineers is a DNA ligase used to glue the human gene. The plasmids programmed by Boehringer Ingelheim are highly efficient vehicles used to insert the genetic information for construction of a human protein into the genome of a cell culture. The cells then pass on the human gene with each cell division and begin to produce the therapeutically active protein. For the synthesis of the required protein, the genetic information is transcribed into a messenger RNA. This messenger RNA attaches itself to a ribosome. There, the amino acids are linked together according to the genetic information. In this way, cell cultures can produce human proteins. Boehringer Ingelheim started genetic engineering of biopharmaceuticals with a range of interferons. These belong to the signal substances which transfer information between cells. Interferons have already been used successfully to treat certain types of cancer, hepatitis, and multiple sclerosis. One of the success stories in this field was interferon gamma, which activates macrophages. Immune stimulation for fighting bacteria which may infect a patient whose immune system has been damaged by chronic granulocytomatosis. In 1985, Boehringer Ingelheim was successful with a further pioneering achievement. Mammalian cells produce the tissue plasminogen activator TPA, which is mainly used to treat heart attacks. The effect of TPA can easily be demonstrated in a laboratory experiment. A thrombus formed from whole blood is suspended in plasma. TPA is added. Within a few minutes, the thrombus begins to lyse. A U.S. clinical study demonstrates that application of TPA over 90 minutes doubles the chances of survival after myocardial infarction compared with treatment using the previously available thrombolytic agents. Thanks to TPA, this patient survived the attack. His wife recalls his doctor's words. He wouldn't be alive without the new medicine. Thanks to genetic engineering, it is possible today to improve selectively the therapeutic efficacy of protein molecules. The TPA molecule was modified to produce TNK-TPA by exchanging certain amino acids. A large-scale clinical study has demonstrated the advantages of a new therapy. In contrast to time-consuming intravenous infusions, TNK-TPA can be administered during the acute phase of a heart attack by injection. The result of biotechnical research designed to optimize molecules found naturally within the body so that they can better meet therapeutic requirements. Physiological substances can only be manufactured for therapeutic purposes reliably and in the required quantities by means of biotechnology. At its research site in Biberach, Germany, Boehringer Ingelheim has installed the largest fermentation plant for genetically programmed cell cultures in Europe. Biotechnology has a long history at Boehringer Ingelheim, dating back to the 19th century. Some of the first products were organic acids and enzymes. These substances were manufactured by microorganisms using classical biotechnical procedures. Since the early days, alkaloids have been isolated from plants by means of extraction, another traditional field of Boehringer Ingelheim. In addition to morphine, codeine, and ergot alkaloids, scopolamine, atropine, and theobromine were also derived by extraction of alkaloids from plants after seasonal harvest. It was thus another revolutionary step forward when it first became possible to use biotechnical procedures to produce alkaloids with the help of fungal cultures, independently of seasonal cultivation and harvest of the plants. Fungal cultures were also important for antibiotic screening. In this field, Boehringer Ingelheim discovered new basic structures by means of its biotechnical processes. Experience gained in the field of classical biotechnology involving fermentation using genetically modified microorganisms, as well as small-scale cell cultures in laboratories, provided the essential basis for large-scale manufacture of human proteins which could then be used for therapeutic purposes. The manufacture of biopharmaceuticals by means of fermentation of cell cultures requires great care, a completely sterile environment, and strict control of temperature, pH values, oxygen, osmolarity, and nutrient supply. All biopharmaceuticals available at present are manufactured without risk to the health of the employees, the population at large, or the environment. In the case of biologicals which previously had to be produced from natural sources, such as blood plasma, urine, or tissue, there was always a risk of contamination, and hence the patient becoming infected with hepatitis, Creutzfeldt-Jakob syndrome, or the AIDS virus. Genetically engineered substances, like those produced by Boehringer Ingelheim, exclude such risks. They have the stamp of high purity and safety. When genetic engineering is applied in research with highly infectious microorganisms, as here at the Institute of Tropical Medicine in Hamburg, it is obvious that strict safety procedures are observed. To date, because of the high standards of safety, no accident or case of illness has been reported. Boehringer Ingelheim can look back over more than 20 years of experience in the fields of biotechnical research and manufacture. All the company's plants operate in accordance with international standards. They are also used for contract development and manufacture. Here are a few examples of successful cooperation with renowned pharmaceutical companies. Babies born prematurely or with weak respiratory organs are often at risk due to a life-threatening virus infection of the respiratory tract. By administering a biopharmaceutical product, it is possible to strengthen their resistance for 30 days at a time. Another example, with the use of interferon beta, it has become possible to reduce the painful episodes of multiple sclerosis. The attacks are now less frequent and considerably shorter.
Vor einem Jahr wäre das noch undenkbar gewesen.
A year ago, it would have been unthinkable.
Seit der neuen Behandlung.
Since starting the new treatment, she's a completely new person. For 15 years, this patient has suffered from rheumatoid arthritis. She has tried all the drugs available, but the side effects always outweighed the relief. Then her doctor suggested that she try the new TNF and IL-1 blocker. That was 4 weeks ago. Now, for the first time in 15 years, she can move again normally. Her dog, who has never experienced her like this before, is just as amazed as her friends. Thousands of other patients have also enjoyed similar experience with the new biopharmaceutical.
Oh, come on, Rex. You really are a spoiled sport.
These cancer cells were removed from a patient with a melanoma in order to test a new therapy concept developed by Boehringer Ingelheim. They are genetically programmed with instructions for the synthesis of interleukin-2 inserted into their genome using plasmids. After that, the cancer cells, thus genetically programmed and rendered incapable of division by means of radiation, are reinserted into the patient's body. These same cells now start to produce interleukin-2, which attracts the killer cells of the immune system. Under the microscope, the effect can be observed. Green are the genetically programmed melanoma cells, and red, the cells belonging to the immune system. They destroy the genetically programmed cancer cells and devour them. The memory effect ensures that the untreated cancer cells will also be destroyed. On the surface of tumor cells, there are proteins which show antigen properties. Biotechnology makes it possible to manufacture tailor-made antibodies. If these antibodies are labeled with a radioactive substance and introduced into the patient's body, they will find the antigen receptors on the surface of the tumor and block them. By means of irradiation using technetium, the tumor can be diagnosed. Or it can be destroyed by means of specifically targeted radiation with yttrium. This is a considerable improvement on radiation from an external source. In the battle against tumors and the control of immune reactions, tumor necrosis factor, another of the body's naturally occurring proteins, plays a decisive role. Boehringer Ingelheim produces tumor necrosis factor alpha, which promises to provide a new form of therapy for patients suffering from soft tissue sarcoma. The walls of the blood vessels which supply the tumor with nutrients and thus permit it to grow are destroyed by TNF-alpha. Blood seeps out within the cancer tissue, coagulates, and blocks the blood vessels. The tumor is no longer supplied with blood and dies. Extremities which would otherwise have had to be amputated can be saved by this treatment in 80% of cases. Since some diseases can be traced back to genetic defects, it follows that it may also be possible to treat them successfully by means of gene therapy. The essential prerequisite is that the gene responsible for the disease is identified. All these examples show that biotechnical procedures are indispensable today if innovative methods of treatment are to be discovered. The Human Genome Project, which is now being pursued by various research institutes throughout the world, aims to have decoded man's entire inherited information by the early years of the 21st century. With 3 billion base pairs, man has one of the most comprehensive genomes of all living organisms. In the future, it might be possible to call up the nucleotide sequence of each human gene from a databank. This will doubtless prove to be a quantum leap in understanding the human organism and its diseases. Even before this project is complete, the results which have been achieved to date can be used. At Boehringer Ingelheim, biochips hardly larger than a fingernail are employed. Their surfaces are packed with up to 240,000 known segments of DNA. These sensors can be used to select, quickly and accurately, specific counterparts from a mixture of countless DNA molecules. This means that in the foreseeable future, there is a realistic chance of gaining a better understanding of diseases for which presently no sufficient therapy is available. New goals, new challenges, which Boehringer Ingelheim will continue to face in the future through the same persistence it has shown in the past.